acids are considered a direct liver function test, but bile acids actually have an actual function in the body, and that's, of course, to degrade fat. And so a few things about bile acids. They're made in hepatocytes, although most bile acids are recirculated, so you only have to make around 5% of bile acids a day. But how they're actually made is from cholesterol. So you get cholesterol from blood, and you get bile acids from blood since it's recirculated, and you have conjugation that occurs, right? The magic of conjugation. And then it's excreted against a concentration gradient. So here's bile acids being excreted into bile canaliculi, and then they're taken along the bile duct, and they go along the bile canaliculi into your bile duct, and eventually they flow, of course, into your gallbladder. They're secreted into your intestine, where they work with things made by your pancreas, which you'll learn more about what the pancreas does. And they break down fats. So that's essentially what bile acids do. Most bile acids are recirculated. So bile acids wind up in your intestine, and then the portal vein via the kind of splanchnic blood vessels, they take up all of those bile acids. They go back to the liver, taken up again in the blood, and then they get secreted, and then that whole cycle starts over again. So recycling of bile acids is around 95% of how we get bile acids, and then you produce around 5% of bile acids. So you don't produce a whole lot of bile acids, but you have to recirculate, recycle, and conjugate the bile acids, which is why it's a great hepatic function test, because it essentially tests both the ability of the liver, the circulature, the vasculature that's taking up the bile acids from the intestine, and how the hepatocytes are actually functioning to conjugate, to uptake them from the blood, so that's a big thing, to conjugate them and excrete them. So again, this is a great direct hepatic liver function test. So what we look at is we actually look at why bile acids increase. So I'm going to present this opposite of how it's in the notes, because I think this is the easiest one to understand. So if you have obstructive cholestasis, where your bile duct, where your bile canaliculi or something in your bile kind of transport system is blocked, bile acids are going to get stuck in hepatocytes, and then they're going to spill over into the sinusoids, and they're going to wind up going into your central vein, and so that will increase bile acids. So that means that if you have evidence of obstructive cholestasis, so increases in bilirubin with increases in ALK Phos and GGT, then you're not going to want to do a bile acids test, because you know it's going to be increased. So the other cause is decreased clearance of bile acids by the liver, and let's talk about that next. So I think the easiest one to realize is a portosystemic shunt, or altered blood flow. So a portosystemic shunt, we see an animal who has, instead of the portal vein going to the liver, it actually goes directly to the heart. And so the bile acids that are in the actual intestine don't go to the liver, they go to the heart. And so that's considered a portosystemic shunt. And so these tend to be young animals, although older animals as well. And you'll see increases in bile acids, and then we'll talk about why you'll see increases in ammonia as well. And they also have a decrease MCV, which is not known why, but they don't always. They may also have decreases in urea, which I'll mention when we talk about ammonia. So portosystemic shunt is one possibility. And then the other one is hepatic failure. And I think this is hard to understand a little bit, so I'm going to try and explain it. So hepatic failure, remember that the liver does not have to make bile acids. It has to pull them out of circulation. So if, here's circulation and bile acids are in the blood. Well, they're really mostly coming from the portal vein. So they then come into the blood and go to hepatocytes. The hepatocytes actually have to uptake those bile acids and extract them from blood, one, and then you have to conjugate them. And if that doesn't happen, they're going to spill back over and wind up. So it's a very active process, extracting bile acids from the blood so that you can actually clear them and excrete them back into the intestines. So hepatic failure would be, again, the other cause because you have decreased sort of clearance of of bile acids by the failing liver. One last thing about bile acids that's important to realize is that these guys emulsify fat. So if you have an obstruction in your bile canaliculi where your bile acids are being kind of regurgitated from here and they're winding up kind of in this area of hepatocytes, they can actually hurt your hepatocyte membranes and digest them, and that can actually result in hepatocyte injury, so increases in ALT and AST potentially. We've talked about indirect liver function tests with hepatic failure, and so you'll need to look for those changes, so decreases in urea, glucose, cholesterol, and albumin. We would expect those all to be decreased with hepatic failure. And remember, those are our indirect liver function tests. So in this case, bile acids being our direct liver function test, these being decreased might tell us to go ahead and do a bile acid test. How you actually do the procedure is you fast an animal and you take the sample, and then you feed the animal and take the sample 2 hours later. So essentially, that fasted sample is just providing kind of an early or really baseline look at the amount of bile acids that's escaping the circulation between the liver and the intestines. But after a meal, when your gallbladder, not in horses, excretes bile acids into the intestine, you're going to expect some increase in bile acids as it is excreted and then taken back up. So you expect a small increase, and you always compare it to a reference range. So that's why we're always going to see that postprandial values are going to have a little bit higher, and that's because those bile acids are being released secondary to eating, and that's an appropriate response.